Welcome, one and all. I'm very happy that everybody's been able to join us as part of a long tradition here in Cambridge. My name is Sam Lipson. I'm the Senior Director of Environmental Health for the Cambridge Public Health Department, but I'm also the chair of the Cambridge Biosafety Committee, which meets monthly throughout the year. This forum is part of a long tradition, as I mentioned, going back to the 80s and before my time. But since 2002, we've been pulling together to conduct trainings to provide background on the relevant science topics and biosafety topics to help support community members who are serving on local biotech IBCs. This is a requirement in our local ordinance in Cambridge. It's a requirement in the NIH guidelines to have community reps. And most cities and towns that have adopted biosafety rules, if not all, also have a community representation requirement. So this collaboration that has been pulled together here really is a classic public-private partnership. We have an enormous amount of support from local academic institutions, from consulting firms, and from other health departments. So I want to make sure to thank the people who were involved in helping pull this together. And let me make sure to get to my my next thank you slide. And that, of course, is the Cambridge Public Health Department. The Environmental Health and Engineering was our primary partner in staging this year. In the past, State Safety Partners has been our primary partner, but they're also assisting us in this forum. The New England Biosafety Association, NEPSA, the Mass Environmental Health Association, Mass Health Officers Association. We have invited current and past and future IBC community representatives who've served not only as Cambridge residents, but from many cities and towns around the Boston area. And finally, I just want to thank and introduce our presenters, Karen Byers, who is from, from hospital. I'll let her introduce herself, so I'm blanking on her affiliation. Melissa Cardwell is a biosafety officer at Harvard, and we have Mary Corrigan, John Kalinowski, and Terry Smith on our community rep panel, which comes later in this presentation. So a couple of things to note before we get started with Karen's presentation. All participants are on mute. If you have a question you would like to ask at any point during the presentation, you can use the Q&A button on Zoom. They won't be addressed immediately. We'll have time at the end of each talk to address a certain number of questions. We don't have all the time in the world. So the session is being recorded, and that link will be made available on our Cambridge Public Health Department website. If you need CEUs or CMs, we will be posting additional information afterwards on the website as well. If you have not registered for session 2, you need to separately register. That's a separate link, and it won't work next Wednesday. So make sure to go ahead and do that. And finally, as the purpose of all of this is to invite and encourage people to serve as community reps, if anybody after session 1 and session 2 is interested in doing so, they can send their resume to me and you will be prompted to do so. So don't worry about remembering to do that. I will make sure that you do. So with no further ado, I would like to invite Karen Byers to begin her talk on some of the overall framework for understanding biological risk assessment as the IBCs carry out this process, and determining whether or not an appropriate level of containment has been assigned to that work, and whether other aspects of the biosafety program are in fact in good stead. Karen will begin her presentation now. Karen?
Okay, I'm going to talk about risk assessment on an institutional biosafety committee. It's something I'm very passionate about, and I've been on an IBC since 1986. Since 1989, I've been on the IBC of the Dana-Farber Cancer Institute, which is where I work. And I will get right at it, hopefully. So one of the questions that I find I still ask occasionally is, you want a what? With what? And why? And I have heard scientists ask each other the same questions. So I encourage all of you, if you have any questions, to get the context of what the proposed experiment is about. Their first section on any IBC registration will be a lay description section. And typically there'll be an answer like, this is the study of, for example, XYZ pathway in maybe brain cancer, bacteria, cells, whatever. The brain cancer answer might be obvious, but for the other types of studies, not necessarily. So there's no reason not to ask. You could learn that it would lead to a better understanding of whatever, XYZ in the immune system, or how neural pathways work, or a new therapeutic drug target, maybe. It is research and they don't know yet, but they're going to invest a great deal of effort into it. And there's no reason why they can't explain exactly what the effort is. And I can say that the community members on my IBC, hopefully they're listening tonight, I believe that they are, always ask those relevant, pertinent questions. If you haven't seen an IBC registration form before, I can assure you that there is on there a summary of the safety precautions that are planned to be used in the experiment. And the person in charge has signed the registration. So in academia, it would be the principal investigator. In industry, it would be the supervisor of the laboratory. But they do have a plan, and that plan is written in shorthand. The shorthand is called biosafety levels, and it covers work practices, special practices, and facility requirements. And if you're new to this, you'll be relieved to know that we didn't make this up. This has been in place for a very long time, doing risk assessment based on these elements. The NIH guidelines for recombinant DNA or synthetic nucleic acid research is available on the web, and the biosafety level practices and facility requirements, etc., are available for biosafety level 1 through 4, animal facilities. They even have greenhouses, insectaries, large-scale culture, such as you might have in industry when they have fermenters. There's also risk groups for infectious agents in Appendix B, which we're going to get to in a minute. There's also the CDC biosafety in microbiological and biomedical laboratories, which I hope you'll let me call the BMBL from now on. And that 6th edition is also freely available on the web, and it's very comprehensive. It has guidance on every aspect of biosafety, including agent summary statements that describe laboratory acquired infections.
Hello, Karen.
Karen, I just wanted to
Yes?
make sure that you share your slides.
I did, didn't I?
Not yet.
Did that work?
Perfect.
Okay. So this was about asking what and why, which I think I went over. And I did say that IBC registrations are going to be signed. So this is where they're available on the web, and this is going to be recorded, so you'll be able to see it. And these are long-standing, generally well-accepted risk assessments for some types of experiments that typically come up in research settings. We're going to talk about biosafety level 1 and biosafety level 2 tonight. Biosafety level 3 obviously has a great deal more detail associated with it, so we wouldn't be able to get through it all. But here are some typical examples of experiments that are done at biosafety level 1. We might use E. coli K12 or its derivatives to express various proteins that are needed for studies. E. coli K12 is a strain of E. coli that is non-pathogenic. In other words, it does not cause disease. And it's typically used at biosafety level 1 and often even in high school laboratories today. Work with uninfected mice would be typically done at biosafety level 1. You might also do cell culture with careful, sterile procedures for the sake of not contaminating your cultures with cell lines of non-human or non-monkey origin, very commonly done at biosafety level 1. Work with fixed material. Again, the fixatives such as formaldehyde would have inactivated the biological components of the tissue, for instance, that you might be working with. However, that is a different risk involved with the particular fixative, but not the biological elements. Where I work at Dana-Farber Cancer Institute, almost every laboratory works with human samples. They make cultures from primary human samples of donated, anonymized samples that are provided by the wonderful community. The blood unfixed tissue would be handled at biosafety level 2. We're aware of the fact that the cells may have a potential infection in them. And so they're handled carefully at biosafety level 2. We also handle carefully cell lines of human or monkey origin because, after all, we're in that same, quote, host range. So if there's something hiding in there that could potentially infect us, then we definitely want to make sure that we use what's called universal precautions or body substance isolation, or in other words, I'm not going to get this stuff in me or on me. Experiments using viral vectors that can infect human cells. Marissa will tell you a great deal more about that, but that is another type of experiment we would do at biosafety level 2. And certain pathogens can actually be grown in biosafety level 2 laboratories. I used as an example herpes simplex 1, the cold sore virus, maybe Epstein-Barr virus, that type of culture. So if we want to do a risk experiment, risk assessment of a new experiment, certainly we want to couple the risk assessment with risk management so that the problem is greatly minimized. So the CDC, the BMBL has pages 10 to 19, 6 steps for how you go through this, identify the hazards, identify the lab procedure hazards, determine the appropriate biosafety level, do a risk assessment review, which is what we're going to do with the IBC. And if it's something totally unfamiliar with all of the members of your IBC, you might call in a subject matter expert, which is something I've done a few times, and I'm pleased to report that I've never had anyone turn me down. Evaluate proficiency, which we will talk about, and revisit the risk assessment and revise as necessary. So we'll get started. Step 1 is to identify the hazard, the biological hazard. So we want to know what bug or virus or whatever is involved in this experiment. The CDC's new term for this is inherent risk. I kind of like it. It means it is the microorganism out there in the real world or in the laboratory uncontained. So as I mentioned, there is a summary in the BMBL, which is presented on this slide, but Appendix B of the NIH guidelines has these risk groups, and they're in 4 categories. So risk group 1 has agents that are not associated with disease in healthy adult humans, and there's a list. Risk group 2, agents associated with human disease that is rarely serious and for which preventive or therapeutic interventions are often available, which is the category of experiments I was talking about a second ago that are handled at biosafety level 2. Risk groups and biosafety levels are not exactly identical, but they are correlated. And risk group 3, agents with serious or lethal human disease that preventive or therapeutic interventions may be available. So there's high individual risk for the person who wants to work with it, but low community risk in terms of transmission in the general community. And risk group 4 is obviously high individual risk and high community risk, and has very limited number of risk group 4 laboratory, very limited number of biosafety level 4 laboratories capable of handling risk group 4 agents. Okay, sorry, said that. Sources of information about laboratory acquired infections. There are many, and it is what your biosafety professional is very worried about. The BMBL has agent summary statements. And so, for instance, if you want to look up what to be concerned about with HIV, you need to go to their retrovirus agent summary statement. And it will tell you everything about HIV, SIV, HTLV1, HVL2, etc. So it is a wonderful resource. However, sometimes what you need is just a safety data sheet for the specific agent that you're working with. So for instance, if I have a lab that's working with HSV1, which is shorthand for herpes simplex type 1, I would go to the Canadian pathogen safety data sheets because they have done a great job of summarizing the disease, how it's transmitted in the laboratory, what has happened in the laboratory in terms of laboratory acquired infections, what disinfectants are appropriate to use, all those really practical details. And I'm sorry, I'm a past president of ABSA. I can't resist plugging the ABSA website, which has a laboratory acquired infection database that is open to the public, and you can put in a specific agent. And if there has been a publication in the scientific literature of a laboratory acquired infection with that agent, it'll summarize it and then give you the reference so that you can get more information. So it's routes of transmission for laboratory acquired infection. So here we're worried about the person and the actual person handling the material. And another reference that's freely available on the web is called MMWR, Mortality and Morbidity Weekly Report. CDC puts it out. There's a supplement volume that is 101 pages that is an analysis of everything that can go wrong and right in a clinical diagnostic lab, which of course all of us are dependent on for any kind of sophisticated medical care. And they find that 20% of the laboratory acquired infections have a definite incident associated with them. There was a needle stick, also called a parenteral exposure, or a sharp, so someone was cut or otherwise hurt with, say, contaminated broken glass. Mucocutaneous, spills and splashes on skin or eyes, nose, or mouth. Those are our mucous membranes. Ingestion or exposure through the mouth, or pipetting or touching mouth or eyes. And then they say 80% are probably the source of the other laboratory acquired infections. So aerosols and droplets, or directly by hand contamination. And I can tell you from years of doing face-to-face training, if you're talking to a large group of people about not touching their eyes, nose, or mouth, somebody inevitably reaches up and starts rubbing their eye. It's just human nature to do that whenever they itch. But it is something that you really can't do in a laboratory setting if your hands have not just been washed. Laboratory procedure hazards we're going to talk about, and at the same time, we're going to talk about how we minimize the biorisk in each of those laboratory procedure hazards. So, you can't see the bottom of it, but this is a device that you stick a tube in the top and it imparts energy to the liquids in the tube and swirls them all around. It's a great mixer. It's called a vortex mixer. And this person, for some reason, has neglected to put a cap on the tube. And I hope you can see that there are some fine droplets spewing out here. I have to thank my colleague at MIT who shared these pictures with me a long time ago. The penetration of the rubber stopper on this type of vial results in some droplets and aerosols. Today, we would also use a safety engineered needle for this step. It wouldn't help us with the droplets and the aerosols, but it would help us with bringing this needle and syringe safely to the sharps container. And pipetting also generates droplets and aerosols. But there is a great fix for this, which is to move all that to the biosafety cabinet. And it's a picture of a typical biosafety level 2 laboratory. This is the device called the biosafety cabinet. So a person wearing a lab coat sticks their lab coat covered arms inside the hood, and they have gloves on their hands. So they are working inside the cabinet, and they have that glass to prevent splash to the eyes, nose, or mouth. And there are air curtains that protect them nicely in terms of their potential exposures. This is all filtered air, both to maintain the sterility of what they're working with down here from the invisible agents that are in everybody's room air. And what's exhausted has been carefully filtered, so no microorganisms come out. It's 99.99% effective. The centrifuge that needs to be used in a laboratory to separate the agents you're working with from the fluid that they were growing in has safety containers so that you can, with rubber gaskets, so you can keep everything inside this bucket and that fits inside the centrifuge. There is an eye wash in a biosafety level 2 laboratory. So you grab it and pull the eye wash, and these little red things pop open and you can rinse both eyes. The OSHA laboratory standard actually included test tube racks as engineering controls. And it's only because lab managers have to keep an eye on whether they're all tied up in refrigerators and freezers and whether there are plenty available for work to hold. test tubes safely. Centrifuging at biosafety level 2. This is a close-up of that wonderful gasket that keeps everything inside. So these are the caps that would go over a smaller centrifuge bucket. And then sometimes, I should have had a picture of a plate. Sometimes small volumes of, say, cells need to be cultured in 96 well plates or 8 well plates, where you can do slightly different experiments in a small volume in that little well in a plate that fits into this device. This is very useful for centrifuging a plate, because you can't screw the cap down on the tube the way you could. It just has a lid, but the gasket keeps everything inside. So at every biosafety level, we focus on sharps precautions, because that is obviously a very preventable laboratory acquired infection. And also it's very efficient because it gets it into us should we have a needle stick. So in any laboratory that you visit, even looking through the door, you'll probably be able to see a sharps container. This is a highly regulated activity to dispose of needles and other sharps, and it goes into these hard-sided containers. This lid is closed. Some of them have a slightly different opening. This is a large one so that you can put in tubing as well as any attached needles, but there are other variations. But the point is, there are sharps containers and they need to be used. We don't, in general, bend, break, resheath, or reuse needles or syringes. We also use safety scalpels with safety covers or retractable blades. And that, again, is extremely helpful when you're going to the sharps container with your used and contaminated scalpel. It does not prevent a slip while you're doing an activity that requires a scalpel. So we need to keep people very aware that when the right hand is holding a scalpel, you need to pay attention to where your left hand is. And you don't want it anywhere near that activity. You want to use forceps to steady whatever it is you're working on. At biosafety level 1 through 3, there's a requirement for a sink. We also check periodically that the researchers have soap available and paper towels, very important in a standard laboratory. We also, in many cases, try to minimize the use of glass as feasible. But of course, certain chemicals would require the use of glass, but not in a biosafety cabinet. So we would not have glass beakers in a biosafety cabinet at biosafety level 2, for instance. Another tremendous help in reducing risk is the use of disinfectants. And whenever you audit laboratories, one place that you always find very clean will be the inside of the biosafety cabinet, because that is always disinfected before anyone starts their experiment and at the end of their experiment, because they don't want cross-contamination in their tissue culture. So that does not tend to be a problem in that area. In terms of disinfectants, again, this is a very highly regulated area. So for instance, even my bathtub cleaner, if I look at the label, it says how great it is to just spray it on and wipe it off to get rid of soap scum. However, if I want to use it as a virucidal product, virucidal or bactericidal, any cidal, those are legal EPA terms, and it tells me how long I'm supposed to leave it on the bathtub, and then I can wipe it off. The same thing is true on any disinfectant product. We tell people the label is the law, and it tells you the concentration and what's called the contact time, how long you have to leave it on. And there's a descending order of resistance. So prions are definitely the toughest to disinfect, and bacterial spores are next, then mycobacteria, and on down the list, which I won't read to you, but I will tell you that OSHA requires, if you're working with blood, that you have a mycobacterial disinfectant, even though the usual concerns that we have for bloodborne pathogens would be hepatitis B, hepatitis C, HIV, which are down much lower here. And the reason for that is that we recognize that the infectious agent might be protected by, for instance, serum if it's a blood sample, or media if it's tissue culture. So there's an extra strong, if you will, choice of disinfectant product just to make sure that the job gets done. And this picture over on the right, I got a 23andMe gift for my birthday. It's a kit that comes in the mail. My daughter gave it to me. I spit into a little tube. I packaged the tube in accordance with every regulation. The packaging was perfect. I sent it to the company. They sent me back a report. I know that they used what's called chiotropic chemicals to get my DNA out of my saliva. That is standard in any type of experiment where you need to look at DNA. And that does very effectively inactivate any germs that I might have had in my saliva that day, or that multiplied in the few days that it took to get through the mail to the company. By the way, they sent me a report back already that says I was related to my daughter, which I knew. And also, this was supposed to be a picture of my chromosome 20, which says I'm not a very good deep sleeper, or at least the sequences of this chromosome is associated with people who are not good deep sleepers, both of which I sort of knew. However, I hope that there'll be more interesting reports in the future. Special practices for laboratories. Well, you can't do biological work without biological waste. And at biosafety level 1 and biosafety level 2, it's very common to send the biological waste off-site for decontamination. And this happens in accordance with Massachusetts regulations, Code of Medical Regulations 480, 000, and also every sort of federal regulation about Department of Transportation for getting it to where it goes. The Massachusetts regulations for biological waste also require that IBCs review the plans annually. And we do. This type of container actually is very promoted by green management because it can be emptied robotically and sanitized and reused. But some labs are growing pathogens, and it is of a level that would be beyond what you find in a typical clinical sample. For instance, if you grow HIV in the laboratory, the quantity of HIV in your laboratory waste is greater than it would be, say, in a clinical diagnostic lab that handles blood. So then you autoclave the bags before you send them out for final disposal. And autoclaves 120 degrees centigrade, very hot. And you choose a time and you teach people on how to use it. You have charts that record how long the autoclave has worked in each of its various cycles. But the point that's of interest to this group is that you validate those autoclaving conditions. So this is how you learn if the time you have chosen is too short for the steam to penetrate to the middle of the bag in a big load, for instance. So the you buy these little vials that are commercially available, and they have media on the bottom, and they also have a little component of spores that were isolated from, believe it or not, hot springs. So these are very heat resistant little spores. You put some of them inside your biohazard bags, and you save a few to be positive controls. So then you put both sets, obviously clearly labeled, into a 56-degree centigrade incubator. And in a day or less, some of them, you will be able to tell that if it looks like it did when it went in, or maybe has caramelized just a little bit, looks like this, you have chosen a long enough time for the cells to be, the waste to be in the autoclave. If your controls look like this, you can see that it has turned yellow. So the little spores have what we call germinated. They have bloomed, if you will, and grown and excreted waste into the media, which has now changed color. So then we know that this group in particular autoclaves all of their waste for an hour and a half because they can't stand it if they don't pass the test, and that we've never failed with an hour and a half.
Hello, Karen.
Yeah.
Hi, this is Lee from Safety Partners, and I'll be moderating the Q&A portion. I just wanted to give a quick reminder that we just have 4 minutes or so just to be able to have time for some Q&A in this session because some questions have been coming in as well.
Terrific. Okay, so things that we can look at to evaluate proficiency: training records, more about training in a minute, the technical training on new equipment. The biosafety cabinets are tested at least annually for compliance with a national standard. The eye washes are drained weekly, and the types of equipment, centrifuges and cell sorters, et cetera, are routinely taken care of. I want to tell you that these are not real pictures. I pulled these off old CDC training CDs. But whenever you train, you want to minimize exposure risk by enhancing awareness of the responsibility for one's own personal safety, such as not playing with your contact lenses in a biosafety level 2 or higher laboratory, but also that the personal safety of the support staff are your responsibility. So something like sharps disposal is key to making sure that the staff who remove the waste from the laboratory are kept safe. Immunocompromised staff may also be at greater risk. You would never ban someone from working in the laboratory due to any of these, but they might certainly want to do a job safety analysis so that certain aspects may be chosen by the individual in consult with OC Health and their physician to be relegated to someone else. And last but not least, revisit as necessary. So reporting of anything that goes wrong, the response should be routine and fact-based, not punitive, because we want to foster those near-miss reports, and we want to involve those people in an effort to improve the program, so to speak. And responses for incidents such as spills should have been developed in advance. It's a good time to go over them again. Okay, I'm looking forward to those questions. Thank you.
Great. Thank you so much, Karen. We're going to see if we can kind of do a little bit of a lightning round here because we did have a good amount of questions come in. Great talk. Really, I learned a lot here myself. Going back to the kind of where we started on the BSL-1, BSL-2 initial conversation, a question on formalin. Specifically, does formalin inactivate all primate and human pathogens?
In some cases, you need to be careful of the ratio. So for instance, with lung tissue, it's 1 to 5 because it has to penetrate through lungs. If you're trying to get through tuberculosis, for instance, that kind of thing. So there are caveats, details, but yes, not prions, but definitely the other biological agents.
Got it. Great. Thank you. And then we had a question come in regards to recommendations of wearing a mask. So would you recommend using a mask to protect mucous membranes for further protection when you're working with human blood and NHP materials? And this is even when working in the biosafety cabinet. Would you recommend a mask for that?
The mask is ideal for splash protection if you buy the correct kind of mask. Some are not good at splash protection, which the market has been flooded with masks lately of many qualities. But a medical mask would be ideal for splash protection. It's not required generally if you're working in the biosafety cabinet, but it might make sense. If there's an incident involved with biosafety cabinet work, it's usually when you're removing too many things at once. You know, you have a stack of plates, you're pulling it out of the cabinet, you hit the side or something like that. I would not work against it, but I think that very specific requirements are appropriate when you actually have the experiment in front of you and you know what the risks are of the potential exposure.
Got it. This question was when we were talking about the risk reduction on work surfaces. So let's say you do not have a designated hand washing sink in the lab. Would a hand sanitizing station work in that case? And if that doesn't work, what would you recommend?
It does work, but you have to get a hand sanitizer that has the appropriate percentage of alcohol in terms of inactivation. And I'm assuming that the individual was wearing gloves and they have paid careful attention to how they removed the gloves so that they did not contaminate their hands when they did that. And then they would move to an area where there is a sink with hand washing available, certainly before eating, drinking, etc.
Thank you, Karen. We had a couple more questions that came in. So just as far as IBC's role in this, you know, the question was, is a role of the IBC to kind of look at labs and observe them while work is in process to see that these recommendations we've been talking about are being followed?
That's not usually feasible, but certainly a walkthrough is appropriate for anything that is new to a particular group. That might make sense, or a presentation to the group about what the procedures are would be appropriate. I don't think you would normally involve an IBC in actually watching operations.
And we have time for one more question that came in. And I'm just going to have this here. The last question is, while using a cabinet, do you know of data in the past, or is there data that you can speak to on exposure while using a cabinet in your knowledge?
Yes, but not a properly set up biosafety cabinet that met NSF 49 certification requirements. There is an exposure to tuberculosis that's described in the laboratory acquired database on the ABSA website. But that was a ducted biosafety cabinet that would not meet any modern standards currently. And in terms of IBC role, I'm still thinking about that question. I just want to add, it's very important. You wouldn't watch exactly, but you are going to be receiving from your biosafety professionals information about laboratory audits and what has been observed and that sort of thing. So you would have a role in reinforcing the appropriate practices.
Great, Karen, thank you so much for your time and also answering these questions from our audience here, and great questions as well. Sam, back to you.
Thank you so much, Lee. I appreciate it. And Karen, thank you. Fantastic talk. Let me apologize, though, for forgetting that you work at Dana-Farber. How could I forget that you work at Dana-Farber? And of course, that you were past ABSA president. So we just have so much privilege around here because we have so many well-trained and experienced people. I want to quickly move on to make sure we don't take any time away from Marissa Cardwell. She is going to be presenting a special topic on emerging risks that are challenging, because the biotech field is constantly evolving. And so it's necessary to keep an eye on those new technologies and new devices and methods that could introduce additional risks. So Marissa, it's all yours.
Thank you so much, Sam. Give me one sec to get my screens figured out here.
It looks good from my side.
Excellent. I'm all set. Well, thank you so much for having me. I am thrilled to be back at the Cambridge Biosafety Forum. So excited that it's happening again this year. I've always found this event to just be really wonderful, really educational. Cambridge has always been a leader in biosafety oversight and fostering responsible and safe research. And this event, in terms of fostering community involvement, is just great. So my talk hopefully will help those of you who are community members or thinking about being community members, just to get a little taste of some of the things that you might see or hear in some of your IBC meetings. Thank you, Karen, for such a great presentation that really sets the stage for a lot of the things I talk about. I don't have to say what biosafety levels are now or speak too much about disinfection. You've covered that. That's great. So I'll give you some general concepts about viral vector risk assessment, and then I'll use a couple different vectors as examples to sort of demonstrate some of the risk assessment concepts. There's way too many details and way too many vectors for me to go into all of them. But just to give you a framework that you can use to do a risk assessment on a viral vector. So viral vectors are very powerful and common tools in biological and basic and clinical research to study gene function. Viruses are naturally good at entering cells. They deliver their nucleic acid to the cell, and they then have the cell use its own machinery to make its proteins and to propagate the virus. This is the whole purpose of the virus, to get in, make copies, and make more of itself. So scientists can actually use that function of a virus and harness that function of gene delivery, nucleic acid delivery to a host cell for their advantage in order to try and study gene function and understand how we can modulate genes and what those genes do. Usually viral vectors are constructed by replacing the wild type viral genes with some sort of transgene of interest. Could be something like a green glowing protein, GFP. It could be something like a gene that maybe leads to cancer. There's a lot of variety, right? And usually we're keeping the useful parts of the virus, and those useful parts are the parts that help deliver nucleic acid. So that's going to be the structural components, the actual, the capsule itself, the receptors that then deliver material into the host cell. And we're going to try and get rid of all the parts we don't need, and especially the parts that cause pathogenicity or infection. We don't want to, have our cells die because we've infected them with a virus. We just want DNA or RNA delivered. Because we're removing so many of those wild type genes, usually vectors become safer than the wild type virus. But we always want to keep the wild type virus in mind when we're doing our risk assessment. That's our starting point. Viral vectors are used in basic research all the time. Again, delivery of genetic components to a cell in cell culture or directly into a research animal. This can lead to expression of a gene of interest, or you could be delivering something that modulates the expression of a gene that's already there in the host cell. Might, ramp up the expression of a gene. It might knock down a gene, just depends on what that payload is within the viral vector. These are viral vectors are also used in gene therapy, and it's been really exciting the last 10 years or so to see the successful usage of viral vectors in gene therapy. A lot of times we're delivering a gene of interest in order to correct a defective gene in a patient, or perhaps we are correcting a deficient gene in a patient's sample that's then delivered back to the patient. And finally, vaccines is another common one. I think we all became more aware of this when the Janssen COVID-19 vaccine came out. That's based on an adenovirus viral vector. And so that's another way that viral vectors are being used. Vector characteristics that affect safety. So these are some of the things that you want to take note of when you're sitting in your IBC meeting. You want to think about replication competency. So as I discussed before, we're removing a lot of the wild type viral genes. So oftentimes vectors become replication incompetent. That's a safety feature, right? We don't want a virus that can get in there, replicate more of itself and spread. But that's not always the case. So it's worth asking to make sure that the vector you're looking at really is replication competent. So don't assume that. Occasionally during the process of making these vectors, there's a potential for reversion to a replication competent virus. So it may be that it's designed to be replication deficient, but something happens in the process of packaging it that leads to replication competent virus. I'll go into all of these in a little bit more detail in future slides. And then, as I mentioned, you want to consider the risk level of the wild type virus. If you get a reversion to replication competency, then you might be looking at the risk of the wild type virus once again. Tropism, what cells or tissues or species can this virus enter? So this is the host range, and that's a huge thing that can be modulated in many viral vectors, and it has a huge bearing on safety. Integration potential, certain viruses actually can take their genetic payload and integrate it into the host genome, and that's going to have different downstream effects. The risks there include insertional mutagenesis. Maybe the gene pops into a tumor suppressor, disrupts the tumor suppressor. You might have a cancerous outcome. The payload or the transgene itself, what have you encoded into this viral vector? What are you delivering? What is its function? And not only what is its function, but how is it regulated? So the gene may get into the cell, but is it going to be expressed? At what level? In what tissue? So those are all really important aspects. A little bit more about replication competency. As we mentioned, we're removing as many viral genes as possible, usually making these vectors replication incompetent. So when the vector gets into the host cell, it doesn't have those genes that it would need to actually replicate produce more virions and spread to the next cell. So that's generally what we want. We want it to get in, deliver its genetic material, but not go any further. But in order to actually make one of these viral vectors, right, we have to somehow package it. It's going to need some of those proteins. And what we do is we use helper function or helper cell. And so in the picture, you see some examples of ways that these vectors are made. The vector is shown in red, and then you can either use a helper cell, which provides those other viral genes in trans, provides them so that you can get viral particles packaged, but the genetic material doesn't end up inside those viral packages. You can have helper constructs, which may just be plasmids. And really the key here is separating the parts you want packaged from all the other pieces of the viral genome that you need to build the viral vector. And that's so that we don't end up having any of those viral genes packaged, and also so we reduce the possibility of recombination. So those pieces sort of finding their way back together and making a virus that can replicate again. Tropism. Tropism is the specificity of a virus for a particular host tissue. It's determined in part by the interaction of the viral surface structure with the host cell surface receptors. In my cartoon picture here, you see that the virus, which is in yellow at the top, has, either circle proteins on its surface, or it has triangles on its surface. And the vector that has circles can only get into the blue cell, and the vector that has triangles can only get into the green cell. So that's really what we're talking about in a very simplified form. So you may hear the terms ecotropic, amphitropic, pantropic. Ecotropic means it's only able to infect, say, one type of cell. And oftentimes we're talking about rodent cells, because oftentimes we're talking about vectors that are specific for rodents. But it just means it can only infect one particular type of, say, species. It's the safest option. So if you have a vector that can only infect mouse cells and you're handling it, well, it can't infect you for the most part. So that's a safety feature. And then amphitropic, it means that the tropism has been expanded to some degree. Now, perhaps it includes infection of most mammalian cells and possibly human cells. So again, now you're going to look at, okay, are the researcher cells, human cells, subject to this virus actually having tropism to their cells and being able to deliver their payload. And pantropic, oftentimes you'll see this with something as VSVG pseudotype. And that means that they've used VSVG, which is a surface protein of the VSV, it's the virus. And this can basically allow the vector to get into just about any cell. It's very promiscuous. It makes a vector incredibly useful in terms of its ability to be used in a variety of experiments. But you do have to also think that, well, just about any tissue of the researcher that gets exposed will be able to be affected by this viral vector. So, and this process of pseudotyping or changing tropism is all about encoding different surface proteins on these viral vectors to change whether they're going to attach to the blue cell or the green cell and enter those cells. Integration. So as I mentioned earlier, there's some viral vectors that can integrate their genome into the host cell, retroviruses for one, so the virus that causes HIV. And this is oftentimes a desirable trait for the researcher because it allows for a long-term expression of a transgene. But there are also risks involved. Insertional mutagenesis, such as oncogenesis, due to disruption of a tumor suppressor, or dysregulation of the host genes that are nearby the integration point. So again, there might be some elements of your payload that turns up the volume, if you will, on neighboring oncogenes, or perhaps suppresses neighboring tumor suppressors, and might then lead to some sort of a mutagenesis. And then mobilization of the vector from the genome of transduced cells. This is theoretically possible that, once the viral vector is integrated into the host genome, at some point in the future, it pops back out and now perhaps, becomes replication competent. You could theoretically have this if, say, someone is exposed to a lentiviral vector. The lentivirus is based on HIV, and then that person is also infected with HIV. That HIV may complement the viral vector exposure and allow that vector to pop back out of the genome. Now, these things are pretty rare, but it's biologically possible, and so it's good to think about them. A very important aspect of viral vector risk assessment is transgene hazard and regulation. So as I mentioned before, you could be expressing a green protein that fluoresces, and we know sort of that protein doesn't have any deleterious effects that we're aware of, considered relatively safe. Or you could be expressing a known oncogene that, if it expresses, very well could lead to cancer. And so those are the kinds of things you want to consider. You also want to consider really exactly how the vector is being assembled and put together. What are the functions associated with the exact pieces that are in there? And what are the other aspects that need to come together to lead to a certain function? So the example I'll give is sort of the CRISPR-Cas9 system. So in order to have the outcome of, DNA is cut, you need to have not only a guide RNA, but you need to have the Cas9 protein. If you deliver both of those things together in a vector, you're going to have that functional outcome of this gene gets cut. If you're only delivering the guide RNA and Cas9 is, say, already produced in the transgenic mouse you're using, or some other way delivered separately, then if you're exposed to just the guide RNA, we don't know that to have a whole lot of function by itself in the absence of Cas9. So it's important to think about all the pieces that are needed for the functional outcome. Where are they? Are they all present? Which ones might the researcher already have present in their body or not? And does that afford you some safety? And then what are the expression control elements? Is the promoter driving high level of expression of your gene? Is it an inducible promoter? So for instance, it's not going to turn on unless you add, X drug, for instance. And so that's a safety mechanism. If a researcher is exposed to that viral vector, but they're not receiving the drug, then we don't expect a high level of expression of whatever that transgene is. So again, this is just the rundown of the topics I just covered. So sort of like the mental checklist to keep with you when you're at an IBC meeting. At the bottom, I've added a couple things such as susceptibility disinfection. So Karen mentioned the variability in the susceptibility to disinfection based on a variety of characteristics. For viruses, you saw the breadth of disinfectant susceptibility. And then another thing to consider is when being used in an animal, is that a permissive host? So is it a host that normally could support, say, wild type virus replication and spread? So sometimes you're going to have a biological barrier there where the animal wouldn't even support wild type infection. And so if you use the vector in the animal, again, it's another layer of safety. We wouldn't expect that animal to ever be able to support a replication competent virus. So now I'll go through a couple of specific examples of viral vectors that you may or may not see. Like I said, there's a lot of them out there. They're getting more complex all the time, but just to give you a little flavor of the variety. So baculovirus, it's a large rod-shaped virus. It's enveloped. Enveloped versus non-enveloped is a good thing to pay attention to when it comes to disinfectants. The host range is invertebrates, usually insects. So generally, this is handled at biosafety level 1. So this is one of those vectors that is a great starting place to look at gene function. If we don't actually have to move into mammalian systems, it's safer to perhaps start here if that's feasible for the research. Even though these are not pathogenic, when they're recombinant, we do still have a responsibility and a requirement to inactivate them, destroy the material, not just put it down the drain. So we still need to consider what the appropriate disinfectants would be. In this case, it's an enveloped virus. So 70% ethanol is perfectly acceptable. Standard autoclaving bleach, there's going to be a lot of things that will be functional against this viral vector. Adenovirus. This is a non-enveloped icosahedral virus. There are 57 serotypes, double-stranded DNA genome. We know this oftentimes as the common cold. So 5 to 10% of upper respiratory infections in children are caused by adenovirus. The fiber proteins on the surface can be modified to change the tropism, so to change the delivery to specific targets. It usually doesn't integrate into the genome. And we generally handle this at BL2. So the wild type adenovirus will be handled at BL2, and the vector oftentimes adapts the same biosafety level as the wild type. So biosafety level 2, we're looking at things like oral contact or viral spread on hands, paper. It's robust in the environment, again, because there's no envelope, and decontamination of surfaces and hand washing. When you think about the wild type virus, it's usually very useful to think about the routes of transmission and the environmental stability of the wild type and apply that to the viral vector. And so that's why you're going to see very similar precautions in place for both the wild type and the vector. Decontamination, because there's no envelope, 70% ethanol is not going to be effective. So we're looking at things like bleach or standard autoclaving. Again, there could be a very long list if you look at the EPA lists that are going to be effective, but these are just some of the sort of basic ones that we see commonly in labs. Adeno-associated virus, AAV. This is a very small, non-enveloped virus, single-stranded DNA genome. It is naturally replication incompetent. So it requires the presence of adenovirus. There's a couple other viruses that can complement this virus and allow it to actually replicate and spread. So it's naturally replication incompetent. It can infect humans, but it's not known to cause any disease. And NIH actually lists this as a risk group 1. So that's very useful. But has a safe profile already. It integrates at a very, very low frequency. Decontamination, 10% bleach or standard autoclaving. I just wanted to mention something about helper virus or helper function and how that could affect biosafety levels of these vectors. So AAV, when used alone, when there's no helper around, a helper was not used in the making of the virus, biosafety level 1 is generally acceptable. If it's administered to animals, ABSL1, and there's no testing for replication competency, it's not a replication competent virus. So I'd say most of the time nowadays, we're looking at AAV being packaged and produced utilizing helper function, helper genes, and not using a helper virus. However, if you did come across a scenario where there was helper virus used in order to complement the packaging of AAV, now you're going to take into account the presence of adenovirus, and the whole experiment gets bumped up to BL2. That is very rare. This is, like I said, kind of gone out of fashion, but I just wanted to mention it to sort of compare and contrast how biosafety levels can shift based on how something is prepared, packaged, and used. I'll draw a little bit of attention to AAV as a gene therapy vector. It's probably one of the more well-known viral vectors in sort of layperson's reading of the news, perhaps. And here's some of the characteristics of it that have led to it being so useful in gene therapy. The wild type virus is non-pathogenic, risk group 1. So that's a great place to start when we're thinking about something that we're going to use in humans and be safe. You can nearly completely remove all the viral coding systems, so that both maximizes the packaging capacity of the vector, the amount of transgene or payload you want to load into it. It also contributes to really low immunogenicity and cytotoxicity in vivo. And so that's really important too. When you administer this to a patient, you don't want them to suddenly react to it. You want their immune system to see it as foreign and fight it. You want it to be able to do its job and deliver the genes. There are some genome limitations due to its small size. There are many, many different serotypes, and that can actually lend itself to some tissue type and cell type specific tropisms. There is persistent expression in post-mitotic cells. So that's also helpful in terms of considering therapy and the need for gene expression. And I just wrote down here that in 2012, there was the first FDA approval for an AAV-based gene therapy drug. So these are being used. Murine retrovirus. These are retroviruses that infect rodents in nature. They are commonly used in research because the rodent is a common lab animal model. They're enveloped. They have an RNA genome. So retroviruses are going to have an RNA genome. They require reverse transcription to DNA, and then they will integrate into the host genome. An example you might see, MMLV, is a common acronym for Maloney-Murine leukemia virus. It infects dividing cells only, which is interesting in terms of its ability to get into the nucleus and integrate. And as we mentioned before, that integration can be really great for long-term expression. It can also cause cancer due to insertional mutagenesis. They come in a variety of tropic flavors: ecotropic, amphotropic, and pantropic. And decontamination consists of 70% ethanol is common. Again, I will compare and contrast the tropisms now and how that can really have an effect on biosafety level. Ecotropic virus. So these are viruses that maintain their specificity for mouse cells. And so they're only going to be able to actually deliver their payload to a mouse cell. Oftentimes they're managed at BSL-1. Again, you have to take into account what they're delivering and a variety of other things, but generally biosafety level 1 is a good place to start. Usually there's not testing for replication competency simply because they can't infect human cells. They can certainly be oncogenic and cause problems for rodents. And when administered to those animals, though, we're really only needing to handle them at ABSL-1. They are the safest option, but they do have research limitations. So as researchers want to establish their research in a mammalian model closer to humans or human cells, there's obviously limitations there. Now, if you take that vector and you pseudotype it, you make it amphotropic, or you pseudotype it with VSVG, now you have a viral vector that should be handled at biosafety level 2. Now this vector can actually get into your cells. It can deliver its payload to your cells. There may be, depending on your IBC, some sort of requirement for replication competency testing. And the use in an animal, depending again on the payload, might be biosafety level 2, might be biosafety level 1. So this one really depends on a lot of the details of what's being delivered. But it just goes to show you that changing tropism can really change the safety profile and how we might think about managing that risk in the lab. Lentivirus. Lentivirus is another retrovirus. It's derived from HIV. It has an RNA genome, and it's able to infect human cells naturally. It can infect both dividing and non-dividing cells, and it readily integrates into the genome. And so certainly with the potential for oncogenic effects. Considering the wild type virus of HIV, we're concerned with direct inoculation, needle stick, entry through wounds, mucosal exposure. There's not a ton of data to suggest respiratory transmission, but we always want to consider aerosols in terms of their ability to contaminate surfaces as well. The lentiviral risks come down to production of replication competent virus, oncogenesis, mobilization of the virus from the genome of transduced cells. And these vectors have become incredibly complex over the years, and for good reason, because a lot of the complexities have continually moved the needle in terms of safety. So I've put a citation at the bottom here. This is a great paper to read if you're trying to do a risk assessment on lentiviral vectors. And this table is adapted from what they have in that paper and just goes through each of the things to consider just for a lentiviral vector. So the vector generation, this is something you might hear in your IBC minute. Is it third generation? Is it fourth generation? What generation is it? And that has to do with how many plasmids are used to actually build this viral vector. The more plasmids means the more separation of all the viral genes. And that means a less chance of recombination and reconstitution of a replication competent virus. Self-inactivation is going to lead to, it's going to help you understand whether there's oncogenesis potential from the insertional mutagenesis, some of the viral genes that are actually inserted into the virus and their effect on neighboring genes. Tropism, we've talked a lot about. Transgene function, we've already discussed. And here's examples of lower risk example, GFP, higher risk example, and oncogene. Expression control elements, how strong or weak is the promoter. The scale, this is something I haven't mentioned yet, but growing up 2 liters has much more splash potential than growing up 5 mils. So another thing to take into consideration, the permissivity of the animal host and the procedures. As Karen mentioned, sharps should definitely be taken into consideration for a viral vector like this. These vectors are often used at biosafety level 2 or 2 with 3 practices, aka BSL2 enhanced, aka BL2 plus. You may hear any of these. They're somewhat interchangeable, but also dependent on the institution to truly define. So make sure you understand what that really means. These are examples of what often constitutes BL2 enhanced, strict use of a biosafety cabinet, elimination of sharps, mucous membrane protection, maybe double gloves in some cases, and lab coats for sure. Decontamination for these vectors, they're enveloped, so 70% ethanol is appropriate, but there's many other disinfectants that have been rated for, say, HIV and can be therefore used for lentivirus. So in general, IBC considerations for viral vectors as you're sitting in your IBC meeting, things to think about. Many of these vectors are available as kits through vendors. This is becoming more and more common. These things are now services that instead of a lab taking a week or more to clone, assemble, package, grow up, concentrate their virus, they use a core facility to create it for them. Or instead of doing the cloning part, they get a kit with the plasmids and boom, they're ready to go. They have a vector in a day or two. So it's important to think about what aspects of the vector the researcher truly knows intimately. Researchers are not necessarily designing every aspect of the vector as they once were. And so you may need to ask your researcher to go pull up the information from that vendor or pull up the information from the core facility to get your questions answered. And then the other part of this is to be confident in the answers to each aspect of the viral risk assessment. Don't make assumptions. Just because everyone else around the table hasn't talked about the tropism doesn't mean it's not important. Bring it up, ask. And, nobody's an expert about all of the things that are going to come across the IBC's desk. It's a matter of just bringing it up for discussion. Somebody in the room will be able to comment on it. And if they seem like they have a reasonable comment and they feel, comfortable with it, then that's a good thing. But just bringing it up is really an important aspect. I've also listed just other viruses that you may see as viral vectors. People are constantly trying to figure out new ways to make viral vectors. If it's a vector that hasn't been well established, it's worth being a little bit conservative before we really understand the aspects of replication competency or what could go wrong. We see certain types of research, say research of the brain and neurons, will tend to use things that are neurotropic, tend to use rabies virus, for instance. And so you may, depending on the IBC you're on and the type of research that comes across the table, you'll start to get more familiar with certain types of viral vectors. So I will end it there, and I'm happy to take questions.
Marissa, this is Lee. There are a lot of great questions. And first off, thank you so much for the discussion here. This has, precipitated many, many questions from our audience, and it's just amazing, amazing content here. So thank you first and foremost. So getting to our questions here, I'm just going to start. Just to confirm, I know you went over lentivirus specifically kind of at the tail end of the presentation there. Just to confirm, would replication incompetent lentivirus be classified as risk 3, the same as wild type?
Right. So wild type HIV, for instance, is assigned risk group 3. You do have to take into account, as Karen said, a risk group doesn't automatically mean BL3. Would the replication incompetent vector be considered risk group 3? I think, in my opinion, if I looked at the definition of risk group 3 and risk group 2, I probably would not consider it risk group 3. And that's why you're seeing it oftentimes classified or being handled at biosafety level 2, because it's a little bit more in line at that point with what we might consider a risk group 2 agent. So there is no official risk group assignment for a lentiviral vector that I'm aware of. So these are things that your IBC has to talk through, but that would be where I land on it.
Okay, thank you. And the question came in, what would you consider the difference, or what is the difference between a gene in trans or a gene in cis?
I really should have better wording for how to describe this, but a gene in trans is generally one that's a gene that's acting sort of from a distance, whereas a gene in cis is one that, maybe on the same molecule of DNA and working within the context of that molecule.
Okay. A question of opinion from the discussion on the viral vector risk assessment. Are the processes for categorizing viral vectors, are they reliable, or do we need to get info about how the categorization was determined?
I guess I'm not sure exactly about what is meant by categorizing. Lee, do you have any feel on when it popped up in the presentation?
Yeah, it popped up. Let's see, kind of in the midpoint there. So it was just when you were discussing the viral vector risk assessment piece.
So
and there's a slide specifically on that. Yeah.
So I'm considering so categorizing the risk group or the variety of items that we take into account. So these items here, perhaps.
That I would go with that.
Okay. So is the process for categorizing reliable? So for going through all of these pieces of information, I think this is about as comprehensive as the folks who I work with in the field have been able to tackle doing a risk assessment for viral vectors. Info about how categorization was determined. So I think that that may depend on how well established a vector is. So for instance, some of these vectors are very well established. There's a lot of publications about their characteristics. For instance, whether it integrates or not, where does it integrate? How does it integrate? So some of those vectors that have been around for a long time, we can be pretty confident in what's there. If it's a new vector, we might not fully know all the molecular details of how it works. And that's where we would want to ask more questions. Do we have published data about that characteristic of the virus? Can the lab even do a test perhaps about replication competency? Don't know if that really answered the question or not. Please, if that person would like to clarify.
They actually did clarify specifically categorizing tropism.
Oh, okay. Are the processes for categorizing viral tropism reliable, or do we need to get information about how categorization was determined? I think once you understand, it is worth asking what specific gene the vector is pseudotyped with. So you could ask, what's the tropism? And the researcher may come back and say ecotropic. But I think it is worth asking, what have you pseudotyped it with, or what is the surface ligand, if you will, on the vector, so that you can confirm that your understanding of the tropism based on that surface protein is the same as the researcher. So.
Great, thank you, Marissa, and thank you, Kay, for providing more clarity on the question there. Appreciate that. Next question would be, in terms of primary intentions here. So is the intention to prevent infection of the person irrespective of whether there is a negative outcome or not?
I'm going to read the person being, say, a researcher handling a vector.
Mm-hmm.
We are always intending to prevent an exposure of researchers to the materials they're working on, especially if they're recombinant. And even though there are murine viruses that we don't anticipate entering human cells, or baculovirus we don't anticipate entering human cells, we would still put precautions in place, albeit at biosafety level 1. Biosafety level 1 is not biosafety level 0. There is still precautions in place at biosafety level 1 to prevent exposure. So these are still recombinant materials. The NIH is still interested in understanding when people are exposed to recombinant material. Sam and Cambridge are still interested in understanding when folks are exposed. And so we still want to prevent that, regardless of whether we think that there is a negative outcome or not. Certainly, if we could expect a negative outcome, we will build upon that and add more layers of protection to truly, truly ensure that we are not going to have any of those what ifs.
Got it. Thank you. Next question is, are tissues and cells homogeneous enough that just knowing generic information about them is enough? Or is there potentially risk introduced by differences between cells that are described as if they were all the same?
So I will talk about this in the context of, I think, risk associated with tissues and cells themselves, and perhaps not necessarily viral vectors. Karen can also jump in if she wants to. So the terms tissue and cells, I would not consider homogeneous enough just by those terms themselves to really understand risk. I'm usually wanting to understand the actual tissue type. Various infectious agents will hang out in specific tissues of the body. And so you're going to want to know what the potential of those agents are for being present in the specific tissue type that you have. So I would get as close as tissue type for sure.
Thank you. This is an interesting question here. So in terms of, so to what extent does the IBC need to consider the spread of viruses and other pathogens to wild or non-human populations? So the example given was a baculovirus escaping into wild insect populations. So, to what extent does the IBC need to consider that spread of the virus?
Yeah, that's a great question. That is a responsibility of the IBC to make sure that we don't have recombinant materials leaving our laboratories. And so you'll find that the NIH guidelines requires that we have appropriate measures to inactivate, kill, whatever word you want to use, recombinant agents such as viral vectors, recombinant microbes, before anything leaves the lab. And so again, this is an example of, it may not cause an actual risk to the researcher handling it. We're still going to make sure that those recombinant materials do not leave the laboratory.
And we have time for a couple questions still here. Still a lot of questions coming through, really active audience. So thanks to our audience and Marissa as well for her great answers here. Another question is, what if the lab is using AAV and an HSV1 or HSV2 used in the same lab, but not within the same experiment? Does that make AAV higher risk, or does the helper virus need to be in the same experiment as AAV?
Great question. So for the whole audience, HSV is one of those helper viruses that might be able to make AAV replicate. So I would not say that necessarily that you'd automatically make AAV BL2 just because the lab works on both of those things. But I would ask the lab what measures are in place to make sure that the AAV that you're working on and the HSV that you're working on are temporally, physically separated. Do you keep them in separate freezers? Do you use separate biosafety cabinets for them? So things like that. How do you prevent cross-contamination? If they've got great answers for all of that, then you may consider that the AAV remains at biosafety level 1. Oftentimes, if you're using one big lab, of course, the whole lab sort of just rises to the highest biosafety level, but really it's about understanding how that lab manages that. If they only have one biosafety cabinet, they keep everything together, and you're not convinced either by their inspection history or the measures they have in place to keep things separate, you may consider raising the biosafety level of AAV. It's hard because an IBC usually wants to have really solid scientific reasoning for assigning a biosafety level. So again, I would just really try and talk with that lab, maybe help them write up an SOP as to how they can keep those things separate, or see if there's another physical space where they can move that work. So.
Great question, followed by a great answer. Sticking with the topic of AAV exposure, so let's talk about during the winter. And so for AAV exposure during winter months, when many AVs are kicking around, should that be taken into consideration, the season, the winter months?
That's a really great question. I will say from my experience in IBCs, the seasonality of various infectious agents is generally, it generally doesn't modulate throughout the season. We sort of just have an expectation that it's going to be there, whether it's winter or summer. But that would be really interesting to look at and understand. I don't have a good answer for that, but it's a great question. So I think the lack of some data with regards to how that affects viral vector competency is lacking, and so we go with a conservative approach. So.
Yeah. And then we had another question coming in during the AAV section. So, an example that was given, cells that are used for AAV for helper functions, they can include HEK 293, but can you mention any additional safety considerations?
For AAV, off the top of my head, I can't think of any. I have not been into the weeds on AAV molecular biology in some time, so I can't think of any others at this moment. It's a pretty simple virus. There's not a lot to it.
Got it. A question on CAR T. Is CAR T considered tumor suppressor?
Ooh, this is where I'm going to need help. I'm definitely not an expert in CAR T. Anyone else on the call?
Karen might be available.
Yeah, Karen. Maybe we can come back to it if we can.
Yep.
Oh,
Here we
is.
go.
Oh.
It is a chimeric antigen receptor on a T cell. So it's a receptor, not a gene that would be considered a tumor suppressor. It is integrated. It is expressed on often cells from an individual, so that it will attack, for instance, their cancer. That there are several FDA-approved CAR T therapies that do that. Does that help?
That absolutely helps. Thank you so much, Karen,
Sure.
for the question assist there. We have an amazing group here. Question on lentiviral use. So for lentiviral use, is additional signage required if the lab space is predominantly BSL-2 seeing that BSL-2 positive conditions are activated?
So the stipulations in terms of working at BL2 plus enhanced with 3 practices, whatever you want to call it, really will come down to your IBC on how you define it. In my experience, most of the time when there is work that is assigned biosafety level 2 plus by the IBC, there is some sort of a requirement that signage is present, at least while the work is going on. It's simply good practice to be able to communicate to others around you. You're doing something slightly higher risk, and they should be aware of that. So.
Thank you. We have a question on COVID-19. So Marissa, do you anticipate multivariate mutations from COVID-19?
I honestly don't have a good answer to that.
It is a novel virus, let's
Yeah.
remember.
Yeah, I honestly, I don't know at this point, and I
Projecting the unknown a little bit. So I wanted to ask an IBC question here. And actually, I think this is something we might get into with our panel a little bit too, about the top 3 roles and responsibilities of an IBC community member, and what are the terms of reference of the appointing town board of health. But I think that we might actually lead into that. Actually, it's a good question. So I think I might ask one more session on the material, Marissa, from this session before we get into our next part of tonight's discussion and panel. Last question here to answer. What do you consider the difference between biosafety level 0 and biosafety level 1 from your practice?
So there are some pretty basic things that we put into place for biosafety level 1. Again, there can be some variations from institution to institution, but the BMBL is a great place to understand what those biosafety level 1 considerations were. Gloves, lab coat, the need to disinfect and clean, absolutely, when you're finishing your work. So that doesn't go away. Biosafety level 1 doesn't alleviate the need for you to clean and disinfect your space when you're done. So those are some of the key things. Making sure PPE stays in the lab. It doesn't go with you. It doesn't, you wash your hands before you leave. You're not doing any of those sort of tasks that would potentially lead to exposure. So no eating, drinking in the lab, no, again, don't put your finger in your contact and play with your contact lens, all those things. Don't apply cosmetics. Those apply at biosafety level 1. Biosafety level 0 is, an office with, a desk where you can do all of those things if you want to. So, so there are some distinct differences.
Great. Thank you so much, Marissa. Great session there. And Sam will conclude with our member roundtable.
Excellent. Thank you, Lee and Marissa. Fantastic. I wish we had another 10 minutes for Q&A, but really awfully helpful. The next section that we intended for this evening's session is really to try to talk people back who are feeling a little overwhelmed. There are a lot of people on this webinar that have a background in some biology or may even be in the biotech sector, and there are a lot of people who do not. Even if you're in a technical field, there's a lot of terminology, there's some of the science, and a way of thinking about how we manage and describe risk that's not too familiar to you, and that's totally fine. So the session we're going to have right now is we're going to talk to a few people who have served. 3 of the 4 have served on biosafety committee, IBCs, for long periods of time, and I think John, one of them, is probably got a little less time, but we really want to talk about the background and the way in which you might manage your role on the IBC. I saw a number of questions come in through the Q&A in both lectures, which were really about how does somebody who's not a biologist or not a scientist play a proper and appropriate role as a community member. So I first want to introduce the people who have joined us to talk about the experiences they've had as IBC community reps. They come from a variety of backgrounds. First, we have Mary Corrigan. Mary is on screen. She works at Harvard, environmental health and safety and industrial hygiene background. Mary is joined by John Kalinowski, who I don't know John as well, so I'll let him introduce himself a little bit, but it's my understanding he's got a little bit less time in serving on an IBC as a community rep. And so we really wanted to have a mix of people, and John was sort of more of our newbie on the newbie end of things. We have Terry Smith as well, and Terry has been serving on IBC community reps for a very long time. And he's on quite a few too. So, but Terry is great to have on this panel because Terry's background is not as a scientist at all. And so I want to kick off by just asking each of the three of you a set of questions. And I'm going to go through, and if you guys can be patient, I'm going to start with Mary and then work to everybody else. And I think if we do this fairly quickly, we'll have some time at the end for you to share any thoughts you want to share on your role, on your strategies, and how to encourage people to serve who don't feel that they are, geniuses in the area of science, but are interested in dedicated community members. So to Mary, the first question, your educational background, professional background, if you could describe that.
Sure. Thanks, Sam, and thanks for having me, and happy to chat with any future Cambridge community members. So by day, I'm a certified industrial hygienist and EH&S professional, and I want to start out with the most important message is that you do not need to be a scientist or a safety professional to do perform this role as a Cambridge community member. So my background is in science and environmental science, and environmental health management, a bachelor's and master's. And as I mentioned, a certified industrial hygienist, and we are trained and certified to evaluate and control various hazards in all kinds of workplaces. And
Great. So let me move on because we're a little short. We gave a little extra time to Marissa. What would you say is the thing you enjoy the most about being an IBC community member?
For me?
Yeah.
I think it's, well, one, it's just, we're so lucky to live in the city that was really the first in the world to establish a requirement for citizen oversight. And so I kind of take that, I don't take that for granted. And I really enjoy hearing about what science people are working on and their therapies. And you'll find when you're on one of these groups that scientists really love talking about their science, and it's really fascinating.
I'm gonna rather than ask you all the questions, I'm gonna move on to John and kind of mix it up a little. John, could you describe your background, professional background, and your your education?
Yeah, sure. Hi, everybody. Thanks for having me. My name is John Kalinowski. I am in the environmental health and safety field, going on about 10 years now. I started out at UMass Lowell in some community health and environmental health program, and I just got an interest in it. And as I grew in my career, I kind of focused more on chemical and industrial hygiene stuff, similar to Mary. And biosafety was something that I always wanted to build on and get to know, being in this area and how popular and how quickly it progresses. So that's basically where I kind of got interested in it, and just expanded my role into that field.
Great, thank you. Terry, to you. Your background, your education. Yeah.
I was an undergraduate major in political science. I have a master's in city planning and a master's in public administration. I've worked a lot in the energy field, which exposed me to some public health issues around removal of asbestos when replacing a heating system, and making sure when you tighten up a house that you don't tighten it up, that you provide enough air changes and things like that back when I was doing those programs. And just sort of picked up things along the way. I started doing this when I was I think it was when I was with the Chamber of Commerce. I don't think I was on an IBC when I worked in either mayor's office. But you spend enough time in Cambridge and you start absorbing a lot of the general information. One of the things I've really enjoyed about this is that it doesn't matter, and this is on the institutional IBCs, it doesn't matter whether you're a graduate student or a postdoc or a PhD student in the humanities or in the sciences. Your professors never want you to die at work. They may want to kill you, but they don't want you to die working in a lab. So they're actually very, very, I've found, and also in the companies that I've worked with, very concerned about their workers in the workplace. And I can tell you, having worked my way through college in things not as clean as a lab, that's a rare thing sometimes.
So I want to give John a chance to answer, because you kind of answered the what do you like, what are some of the things you've enjoyed about the process. John, what would you say are some of the things you've enjoyed from your experiences as a community rep?
Yeah, so as I am definitely new, I'm going on my second year at a kind of a newer, smaller company. So it's been great to break me into it, and it was, pretty much everything I hoped it would be. And really, it was I started expanding the role into my own company, and addressing the issues I had and learning a ton, and being able to now see what other companies were doing and share some of the lessons learned, and actually take some things back to my own day job was great. But also being able to step back and really think, I am a community member, so I do want to make sure that things are being contained there and also at my own place. So it's just great to share lessons learned and just, be in the mix of it, basically.
That's excellent. Thank you. I want to ask Mary a question about what she considers a best practice you've seen on an IBC. And I mean, as it relates to how they've treated you or other community reps.
Yeah, I guess I have been on them for quite a few years, and I've seen quite a few best practices. I think just one is, the first is just this, you immediately get the sense of welcome. They're so grateful that you're serving, and they think I've never met a company that hasn't been. I think the ability to ask questions before or after a meeting, and follow up and really genuinely express interest in what all the members, and I'm usually serving with another community member too. So it's not because I'm a safety person. I really feel that it's a genuine interest in what the members have to say.
I want to ask a final question that goes to all three of you in sequence, and leave a little time for you to share any open thoughts you may have. So this is about what is one thing you think the IBCs you've served on could do better to accommodate the role of the community member? And I'm sorry, to Mary. We'll start with Mary and go to John and finish up with Terry. Yeah.
Yeah, I've been on a lot of these, and only about, a few months ago, actually, I'm also on the Cambridge Biosafety Committee, and one of the companies came forward and they just did such a great job presenting the information about the flow, and they actually showed pictures of the equipment, and we're going to do this and we're going to do this. And I was like, it was like kind of a eureka moment for me. I've been sitting on the, a lot of pros. That was really helpful. I think every institution probably has to remind itself that they really are supposed to be giving a summary in layperson's language. This really isn't the summary that they're giving to their scientific board. And I also think, it's great when they discuss incidents. It kind of shows that you're a partner in problem solving and a level of trust that companies have. And I think, we all can think of accidents or incidents we've had. So I think everyone always has something that they can contribute to that kind of conversation, even if they've never stepped foot in a research lab.
Excellent. Thanks, Mary. John, in your first couple of years, anything you pick up that you think they could do better?
Yeah, so I mean, let's see. I definitely do agree with Mary that the thing about the layperson terms and providing that information just makes you feel a little bit more welcomed and comfortable. And so far, the one I've been on has done pretty good job at that as well. I think just the general idea of just sharing and, maybe even being a little bit more open with other things outside of just the biosafety, like other incidents that maybe occur, just to kind of give you an idea about the overall safety culture at the location, and just how, other things could contribute to potential, biosafety incidents or exposures to staff or their community. If you have these indications that maybe some other areas are lacking, can sometimes show you some signs.
I'm going to throw in one before I go to Terry, which is something about offering, especially new members, a background, a little additional sessions, additional support on the science, maybe the technology platform before they attend their first IBC meeting. I think that companies don't know what people need or want. And if you don't ask for it, they really don't know. So that is something I think they could often do better at. Terry.
Yes, so I was going to mention that that's one of the things I asked for. And that goes back to my first at Whitehead, where I spent a lunch and was they pretty much laid out what, before I was on it, what type of work they were doing there. Lay language is important. Falling into jargon, you need to be willing to say, I don't understand that. Or there's a three initials someplace and you can't find where that's defined in the information you've been given. I wish, not just for the life science companies and the organizations that I'm on an IBC, I wish everybody paid attention to the fact that not everybody knows what an acronym means within the context of their organization, but we all fall into it. And going back to what Mary said, one of the things I've been insisting on, since more of these have been by Zoom than on site, is that they have a visual that lets me know how the flow goes. The first few I was on, I did a walkthrough before I signed on, and I think a walkthrough of the facility is always useful. Not the whole building, but just this is what our lab space looks like. This is on a Tuesday at 2:00 when people are just there doing work and they say, come on over. That to me is a sign of openness that they really want the help, and you can see potential issues.
Here, you actually kind of got at my extra bonus question, even though you didn't know what it was going to be, which is a situation where you didn't know something and you needed to ask. And I'll expand that to like finding a balance. You don't want to interrupt the meeting constantly, but you also need to follow what's going along and to a significant degree. So how do you strike that balance?
I ask, and if I don't know something, I'm going to ask, and if the explanation isn't clear, I'm going to ask again. It hasn't been a problem. People seem to really want to share that information, and sometimes it elicits from the other staff some conversation that may have been lost. I remember one, I won't go through the organization, but there was a researcher who wanted to make their own controlled substance rather than buy it. And I saw this and I said, well, why would you do that? And pretty quickly, everybody else in the room said, yeah, that doesn't make any sense at all. So
you were the one to ask the question. Yeah.
But I was the one to ask the question because I'm the researcher who's sitting there in the room isn't going to be offended if the community person asks the question. Somebody in the room who the person may have to deal with, or may have to deal with the person, may feel a little not sure. So sometimes, asking that question elicits a conversation that may not have occurred. And sometimes you just catch something that nobody else caught, which is fun.
Excellent. So we did get a lot of questions in our Q&A, but I also wanted to give a chance for, I mean, you guys answered my questions brilliantly, but if you have any other thoughts, any open thoughts you have on the role, any pep talk, encouragement to people who haven't served in that role, this would be a great time to share that. So that's to anybody who has something they want to add. Merritt?
Yeah, I'll add that you're really never alone in this role. The committees have a biosafety officer or a safety person, professional that's maybe wearing both hats. So that they're always very happy to talk with you during, before, after a meeting. Sam is a great resource. People can always, you get the materials in advance. You can always, say, read it in advance, ask questions beforehand. And I think, any of us are on this panel would be happy to chat with anybody who wants to, talk more about how we've approached being a community member.
I will make sure to forward the requests to you as they come in, so we don't hand out your email address. So,
right.
Terry.
Yes,
I'd be happy to talk to anyone. And when you start with any organization, I'm on 12. So I have some that I've been on for years and some I've been on for one meeting. The flow, how they do things from the beginning to the end of they give you their research plan, they give you what they're going to do, then how does it, you have them walk you through how that actually works in the space. That's a good way to start understanding the organization and its culture. Over time, the science, the type of research, all those sorts of things, you'll know more. Always assume that they know more than you, because they do, and always be willing to ask a question. Always be willing to ask a question.
So we do, John, you have the privilege, if you want to share anything. We have about 5 minutes to get through more questions than we can probably answer, but I want to give you the privilege.
Yeah, no, I just want to say, I'm an example of someone being new, you know what I mean? And it's I do feel welcomed, and you can always ask the questions, and people, that are doing the science, they also like to talk about it. And once they understand you have an interest in it and you're asking questions, the conversation usually goes in the right direction, which is fantastic. And, it's just generally rewarding to kind of do something. It is kind of related to my day-to-day, but I also get a chance to kind of do something else for the community outside of, my job, which feels good.
Excellent. Thank you. Thank you. All three of you were really helpful, and I hope encouraging to people who haven't yet played in that role. And so I'm going to read through the questions. Lee has been a terrific wrangler for our questions, but we may not have enough time to get through them. So I'm going to read directly. The first question is about how do we find out about near misses? And the reason I decided to include that question to try to answer it is it's sort of about the culture of the lab and the culture of the IBC. If you don't have an actual accident, you had a near miss, that is obviously something that somebody has to voluntarily share. And it's a part of a culture that can be encouraged, and in many companies is encouraged, to use things like near misses not as a punitive opportunity, but as a learning opportunity. And it's a very good sign when you have that being shared, but it's not something, a near miss is not something you can absolutely ensure that you're going to hear about. That's just how it is. So there's this is a more detailed question. I think I want to move on to process. Top 3 roles, responsibilities of an IBC community member, and what are the terms of reference of appointing town board of health? Not sure I understand that second part, but I would say top 3, and then any of the 3 of you could jump in, but really you're there as a reminder to the company that they have a community that they are part of, that they're accountable to. So it's, even if you did not play a major role in that IBC, maybe, it takes a while to get used to asking the right questions. Your very presence there is an important reminder that there are things expected of a company and that they are in fact accountable to the community they're in. So I'd say just be there. And other roles, responsibilities, of course, is to try to force them to say things in plain language so that you can understand them. So even if you do understand some of the technical jargon, it's really valuable for them to have to know how to talk about this with the public at large. And I think that's another role that you play. If anybody else has any thoughts,
Well,
it is,
one of the
yeah.
one of the ways to learn whether, about whether it's near misses or pressures in the organization or whatever is small talk.
Mm.
You show up for the meeting, and before it gets started, you just ask, hey, what's going on here? What's, I see you moved from one floor to another, or there's a new person on the committee or whatever. And you can elicit friendly information. People talk.
Yeah, Mary.
Yeah, and I would say when the companies do discuss their incidents, that's a good opportunity to ask about near misses. But I also use it as an opportunity to remind them how good it is that people are reporting incidents, because I think they especially think a community member is going to be aghast that something ever happens. So it's like, it's a good culture when people are reporting.
So the next question here is about IBC registrations, and should they include what other projects are taking place in the same lab? I think it's about whether it's a regulated protocol or experiment versus other work that's going on. That's how I'm interpreting the question. The way that we talk about it, formally speaking, our ordinance and those in most communities around Massachusetts are related to recombinant DNA and synthetic molecules, synthetic or nucleic acid molecules. So that doesn't mean all biology. That means biology involving manipulation of the genome and genetic material. However, when we talk with labs and I talk with them and try to guide them, I put it this way, that the biosafety program is really kind of an indivisible thing. It's about all the risk is how you manage it, how you identify it, how you mitigate it. And it doesn't make a lot of sense to separate out work, especially if there's work which is potentially infectious, from that work which isn't. So that's how we view it. It's like, that's not a fully functioning IBC if they're not talking about all of the risks that are being posed in the lab. So we are at time. It's 8:05. We grabbed an extra 5 minutes from you all. I'm very happy you could join us, and I really hope people are able to register for the second session next Wednesday. It is a separate link. Keep that in mind. We're going to talk more about some of the management of a biosafety program from the municipal perspective, which I think there's a number of people in this workshop who would like to hear about. And we'll have a mock IBC as well. And I think you'll find it pretty rewarding. So once again, I want to thank all the 24 people who made this possible. And I'm really very excited for next week, and I hope you all join us.